The overall goal of this procedure is to prepare organotypic transverse slices of the forelimb region of GFP-expressing mouse embryos to image spinal nerve outgrowth. This is accomplished by first embedding the embryos in low melting point agarose and limiting the area of tissue and agarose to the region of interest. The agarose block is then mounted for slicing. Then, the embryos embedded in the block are sliced with the vibratome. The final step of the procedure is to remove the agarose surrounding the slices and to transfer the slices into culture plates. The slices are then cultured and show outgrowth of peripheral nerves into the periphery, which can be monitored with fluorescence microscopy.
The main advantage of this technique over existing methods, like a guillotine or a tissue chopper, is that the slices are generated in a more controlled fashion and show improved viability in culture. Therefore, this method can help in the study of developmental neurobiology, such as looking at the guidance and the outgrowth of spinal nerves. Demonstrating the procedure will be Isabel Brachmann.
Begin this procedure by preparing 10 cm tissue culture plates with 6 ml of culture medium and 3 cm millicell cm 0.4 micron culture membrane inserts. Keep them in the incubator at 37 degrees Celsius and 5% carbon dioxide. Next, heat up 4% low melting point agarose in PBS in the microwave and keep it on a heating plate so that it stays at approximately 37 degrees Celsius. Fill 10 cm bacteriological petri dishes with PBS and place them on ice. Then, set up the vibratome cooling device.